Hi everyone, and welcome to the 15th TIA Centre seminar of the academic year. My name is Adam Shepherd, and I am a postdoc here at the TIA Centre. For those that are new to our seminars, we invite researchers and leaders in the area of computational pathology and associated areas to discuss their work. Today, I'm absolutely delighted to introduce Dr. Theodore Zhao. Theodore is a senior applied scientist at Microsoft Health and Life Sciences Research, working on multimodal biomedical imaging models, as well as biomedical natural language processing. Theodore earned his PhD in applied mathematics degrees from University of Washington. His research interests focus on machine learning, self-supervised learning, multimodal models, and mathematical modeling. And the title of Theodore's talk today is Foundation Model for Joint Segmentation, Detection, and Recognition of Biomedical Objects Across 9 Modalities. Theodore, thank you very much for joining us. It's great to have you here. And when you're ready to start, please go for it.
Yeah, thank you, Adam, for the introduction. I'm very honored to be here, and thank you for accommodating the time. I know it's not the regular time, but yeah, that said, let's get started. So I changed the title a little bit, but it's the same thing, just the two for better understanding. So I was describing the model called BiomedParse, which is by Microsoft, and it's a multimodal biomedical image segmentation analysis model. So before I jump into like the technical details, let me just thank all the collaborators. So this is a joint work from a lot of companies and orgs, and including Microsoft Health and Life Sciences Research, and Microsoft Research, and University of Washington, and also the Providence, the hospital that provide clinical insights. And so let me give a overview of the problem first. So in biomedical image analysis, we always ask a lot of questions. So for example, let's say here is a MRI of the brain. We would want to know what's the size of the tumor in the brain and what of the part of the brain is actually necrotic. And sometimes for another, like a chest X-ray, we want to know, is this patient infected by COVID or other diseases, and how much of the lung is getting infected. Is just a small portion or a large portion. And sometimes when we have a pathology image, we want to know, is there any like neoplastic cells in this image? And if there are any like immune cells near the neoplastic cells, that we can do some further analysis. So all of these is kind of like fall into the semantic segmentation region, where you want to associate the regions in the image with some certain medical concepts. So when doing so, there are a lot of different segmentation tasks because, as we know, like biomedical images, they come from multiple modalities like X-ray, CT, MRI pathology, etc. Even let's say for the cell imaging, there are also so many types of modalities that look very, very different from each other. And secondly, there are different targets when you try to do the analysis. Sometimes you want to find where is the organ, sometimes you want to find where is the lesion, sometimes you want to find certain types of cells. So these are all very different. So traditionally, people train multiple specialist model for each certain tasks. So let's say they train one model for detecting tumor in the brain MRI, another model try to find out some certain like histological structure in the H&E slides. Sometimes they want to train another model to find the infected region, so on and so forth. About like 1 to 2 years ago, under the trend of the segment anything model, there was a model called MESM that tried to unify all different modalities and targets into one model. So this one model is able to segment a lot of like different biomedical objects, but you need to provide a bounding box for each object. So let's say for this tumor, you need to provide a tight bounding box around the tumor, and then the model can get the very fine-grained like contour for this tumor. And for this this segmentation, I believe this image actually come from the TIA Center. So the glass data sets. So you need to give a bounding box for each single like structure. and then the model can give you the structure like separately. And the same for, let's say you want to find a tumor, you need to have a very tight bounding box here around this very, very small tumor. So there are a lot of like human efforts in this. So it's not like a one shot and it does everything. The human expert need to first be very precise in locating the object. And then the model is able to do the last mile, getting the pixel level segmentation out. So what we wanted to conquer is try to unify all these different tasks in terms of language. So this is also following the trend of the current large language models. Large language models can do a lot of things. You don't train like separate expert models like in the past, but you have one model. But how can you have it do different tasks? You just by telling it in natural language. Basically tell the model what to do with the medical concepts, and we hope the model can associate certain tasks or like the objects in the image with some like biomedical concepts. So let's say in this pathology image, if we ask for different types of cells, we hope the model can precisely locate and segment out all those different types of cells. And for this like abdomen CT scan, we want just controlling by language input, the model can go for the healthy region of the kidney, like the tumor and the cyst in the kidney. So that's the goal. So by doing so, we remove the effort like in manually providing a precise location, and the model is able to have better understanding across different concepts. So we used this model architecture. So here's just a brief overview. So we build our model upon the theme, which is the segment everything everywhere model architecture. So it takes in a image and a text prompt. They each go through the image encoder and text encoder. And then in the output, we have the mask decoder to have the fine-grained segmentation mask. And there is also like a very coarse grain meta object classifier, which goes into just a handful of different like meta objects. But the precise object is actually controlled by the natural language. So you can describe the things in text prompt, and the model can output much more fine-grained output. So in order to do this task, the model architecture is just the one thing. The most difficult, most challenging part is the data sets. So I'll spend some time describing the data sets we built for this purpose. So at that time, there was no like text to segmentation data sets in the biomedical domain. So we went on and built the first medical text to segmentation data sets. So it contains in total of like almost 7 million image text pairs. And all these data sets, they were derived from 45 public data sets, which includes some pathology data sets from the TIA Center, including GLASS and PanNuke. So yeah, so I'm very excited to see the invitation because I deal with all these data sets and I know where each of them were coming from. And these are very great, very precious input for the model. So in all these data sets, because we know what are the class labels of those types, so we can actually use large language model, let's say like a GPT of some version to get a variation of different description for the text. So let's say we have data sets which contains chest X-ray, and the label for in this data set is COVID-19 infection. And then with this information, we can prompt LM, let's say, hey, I have this task. Can you generate a variety of different prompts for that usually typically a clinician will say and to describe what's there. And then the LMs did a very good job in generating like a variety of text. So all of these, they went into like the training data so that we can build the image and the text pairs based on this variation. And we have open sourced these data sets on Hugging Face. So if you are interested, please welcome to download, and then they are all like from the public data sets. So when we want to do a segmentation task, we need to first define it. So in order to better unify all these different modalities and tasks, we found there are like three components that can be most unique to define a task. That is like how was the image created? That goes to the modality, what technology we're using, and where was the imaging performed. That's the site for the modality. For example, this one is X-ray conducted at chest site. And for, let's say, you have a pathology image, it can be, so the modality can be like a H&E, and the site actually can be the tissue. Where is this tissue coming from? It's come from kidney, or it's come from like adrenal gland. And lastly, this is most importantly, what's the target to segment? So in this example is the infection that's in this chest X-ray. And alternatively, let's say you have a pathology image, you can say, I want to find like a nucleus in some like a breast, like pathology slides, something like that. So we found like this triple definition to be very uniquely defining a certain task, and is very broadly applicable to many different tasks. So we use this definition to combine all of them, and this is the prompts, the input to prompt the large language model to generate a variety of descriptions. I saw in the chat there is a question.
Hi, Theodore.
Okay.
Yeah, we've got a question from Pingjun Chen, who's asked, in the SEAM architecture, how do you fuse the image encoding and text encoding for master decoder and meta-object classifier?
Yeah. So yeah, this is a very good question. So I think I can go to more details in the following. So basically so from a high level, this mass encoder is a transformer that's taking input from the text embedding and image embedding. So this transformer will cross-attend to the image embedding in some masked manner and do self-attention with the text embedding. So they go iteratively. Let's say cross-attend to the image and then self-attend with the text, and cross-attend to the image, self-attend to the text. So that's how it works. I have like a more detailed but that's the updated architecture in the later slides, which we can go into more depth.
Perfect. Thank you very much, Theodore. I hope that's answered your question, Pingjun. And I think there's also a comment. I don't know, Theodore, if you were trying to show your mouse or not, but I don't think we can see your mouse if you're using it. I don't know if you can put it onto laser mode or something.
Let's see. Laser pointer. Okay. Can you see the mouse?
Yes, we can see it now. Thanks for sorting that out.
Yeah. Okay.
Brilliant.
Let's okay, so we collected many different data sets and unified them in such way. And this collection has a very diversified like a modality and size computation. So in total we collected 9 different modalities from like CT, MRI to like OCT. And some of the data set, as you can see, they have very large like a percentage in the total collection, because in the community there's just so many abundant like a CT and MRI of so many different tasks. But some, let's say like a dermoscopy or endoscopy, OCT, they are kind of like a rare in the domain, and the task is very limited. But anyway, in total, we collected like 9 different modalities. And pathology, the size is the size is good thanks to TIA Center, but we also found like a more fine-grained annotation to be very expensive. So I think that's also something that in the future that hope we can tackle. And this variety also cover a lot of like different sites conducted in each modality. For example, CT, we have that conducted at abdomen and let's say chest, lung, so many different sites, and the same for other modalities. So we collected also a variety of anatomies and object types. So all of them, each of them is described by a text prompt rather than predefined class. So we don't need to, let's say, set a model, say, hey, you output like at least 100 classes, then that's fixed. But rather we control these different types by different text prompts. So we prompt the model with different in natural language, and the model can output the different like binary segmentation mask. And this way we can make it very extendable to new object types. Is there other questions?
Thanks, Theodore. Yeah, we've just had a question also from Craig, who's asked, with respect to pathology count, is this number of whole slide images or is it related to patches?
It's patches. It's patches. Yeah, that's something I also cover. So currently this model is not able to do whole slide image. That is very big and hard for the model to handle that high resolution and the large scale. So what we did was cropping into very small crops and then process each one and iteratively go through the whole image. Yeah, that's a very good question.
Sure. Thanks, Theodore.
Yeah, so here are some like some of the examples from the so-called like input and output pair. So we have a text prompt describing the modality and object types, and the output is the corresponding segmentation mask. So this covers like a variety of like 9 different modalities. Okay, so now I go into some like a quantitative results and analysis. To learn more about like what's the performance of the model. This is okay. So here is like an overall performance comparison. So we compared our model like in the BiomedParse with like a bounding box prompts like MetSam and SAM. So for these 2 models, for each of the object, we provide a precise bounding box around it. And we also compare some like text to box prompting, which we first use a text to bounding box model called the grounding dino to generate the bounding box, and then use this bounding box for the MetSam or SAM to conduct the fine-grained segmentation. So we found that with pure text prompting, we actually outperform those models that got the Oracle bounding box, which is a bit surprising to us. But I will go into more detail that why that's even possible. And overall, this advantage is like uniform across like different modalities. And for the text to bounding box prompts, this is suffers a lot and is not getting a very good performance because the grounding dino is actually lack the knowledge from the biomedical domain. So the bounding box it gives is very noisy, and the downstream model is very relying on the very precise prediction of the box for it to conduct the segmentation. So we also did a comparison with like expert models. So instead of like having one model doing all the tasks, we train expert models to do each specific task. So in total, for each model, we train like 95 different expert models, and to see what's the performance of this model performing on this specific task, and then compare with like a BiomedParse, which is one model conducting all the tasks. We found that the BiomedParse is on par, at least on par with the NNUNet and much better than the DeepLab V3. And NNUNet is a very strong baseline as a supervised segmentation model, but the performance is like almost the same for most of them, and some of them significantly outperforming. So we also compared the model with other like text to segmentation models in 3D, including SegVol and SAT. So because these two they're only like for the CT scans. We just compared like different organs in the CT segmentation, and we also found like the BiomedParse is outperforming like these two 3D models. So now go into like why this is like a, let's say we have a text prompting model, it's even performing better than providing a tight bounding box. And the reason we think is text is much more expressive. So let's think of like how you can define a bounding box in terms of like numerical numbers. You need probably 4 numbers to define it because of the coordinates in X and Y. You can precisely define a bounding box in a 4-dimensional vector. However, in a typical text embedding or text embedding models we are using, the dimension of the text embedding is around like a 512. So that's like hundreds of dimensions. So that means the text contains much more information than a tight bounding box. So let's go to some like more concrete examples. So let's say I want to segment the whole tumor core, which is like this like orange region. For BiomedParse in text, we can say, hey, like let's go for the tumor core, and the model can do that. Okay, for the bounding box model, this corresponds to this very tight bounding box around the tumor core, and the model can do that. But let's say we want to only segment the enhancing part of the tumor, which is like this outer region of the tumor. In text, you can control it precisely by changing the language. But you will find out that the bounding box is exactly the same. And for this type of model, there's no way you can distinguish like these 2 different tasks because they have the same input. You cannot expect the model to magically output precisely another like a segmentation mask. And the same thing happens with, let's say another example. This is the prostate peripheral zone, and the bounding box is like this. But you cannot go into more details, let's say, because the model will think this is the bounding box corresponding to the whole thing rather than this like outer region. So mathematically, this is because the dimension of a bounding box is too small to be able to capture all those like different possible region of interest. But with text prompting, it's totally possible because of the dimension is just so large. The expressiveness of language is very much higher than bounding box. Another example is when you want to segment like a liver vessels, that's like these little spots scattered in the liver. If you just have a whole bounding box, the model is confused and do you want the whole region or how can I know I just want these? So that's a very like this is a concrete example. So if you look at all these shapes, you will find out these shapes are kind of like irregular. For example, it's not like a convex shape. It has a weird shape or scattered around. So we went down and performed a analysis about what's our model advantage versus the irregularity of the shape. So we define like 3 different metrics. The convex ratio, which is define how convex the shape is, and the box ratio, which is what's the area of the shape divided by the area of the tight bounding box. And lastly, is like the inverse rotational inertia is describing how spread out is this shape. So the higher these indices, the less irregular it is. So for example, the box ratio when it goes to 1, that means this is precisely a bounding box. And the same thing for this like convex ratio when this shape is exactly convex and this ratio goes to 1. So we found that the model advantage on the y-axis goes down when this shape gets more regular. So when this shape is like exactly convex or exactly like a bounding box, so our model even is like underperforming those like bounding box model, which is like understandable because controlling by bounding box is much more accurate here. But for most of the case, those shapes can be weird. And in that region, our model performed like much better than this like a bounding box controlled method. Okay. So we further conducted some analysis of the language encoder trained with our model. So we chose like four different categories describing four different types of cells. So for each category, for example, like epithelial cells, there are a bunch of like different prompts describing it as generated by the large language model, and these cells can also resides in different like tissues and organs. So we plotted the text, the language embedding of all these like different types of prompts, and we found like our model is able to separate like different cell types very nicely. In comparison, we also tried like the BiomedBird and BiomedClip imaging text encoder and found its capability to separate the different cell types is not as good as our model trained to do that task. Okay.
Right, Theodore, do you mind if we just ask a quick question there? Mustafa, sorry.
Yes, just a quick question. Did you experience the same behavior? No, in your next slide.
Experience what? Sorry?
Did you experience the same behavior in your next slide when you work with other modalities, or was it just for the cells?
You mean like this slides?
Yeah.
Oh yeah. So for cells, because there are many different tissues and we have a good collection for each cell type, we have about 100 different descriptions. For other modalities, usually like one target, it contains just a handful of like variations. So it's hard for us to conduct like this clustering visualization. But I think the behavior is the same. So we just use this as one of the examples.
I see that. Okay, I see the reason. The only reason I asked was it because the data that BiomedBird or BiomedClip was trained on, because they use less or no pathology images, as far as I know. I'm not sure. So I was just wondering if that was the case, or.
So BiomedBird, so the BiomedClip, they froze the text encoder of BiomedBird. BiomedBird was trained on the PubMed dataset. It's like a language only. So this model never see any image and try to associate certain things with an image. In some sense, it's trained in a self-supervised manner, that the bird style, just predicting the mask, the token. But our model is actually training a supervised manner. So because for this type of cells, it needs to conduct the segmentation, although they may look different in different tissues, but somehow the model can try to associate what are the image feature can be like an anchor point for the text model. So I think that's the reason for this.
Yeah, cool. Thank you.
Yeah.
Thanks, Theodore.
Okay, thank you. Okay, so now I'm trying to see like the capability of this model in handling like different tasks. And for side notes, we have the model on Hugging Face, and we also have a project page to describe the model. So with this text prompting, we can do things like having one prompt and have it process all the images. So let's say you're a hospital and you have a lot of like CT scans for lung, and you want to know where, if any of them have lung nodule, and where is the lung nodule. So with text prompting, you can apply the same prompts, lung nodule in CT, to all the images, and then model just goes on its own and segment out all the lung nodules. So that's like one shot, and then you have everything. So if you want to work with like a bounding box type of models, you need to find the nodule in every CT scan and then provide a bounding box. So that makes it like a much more laborious. And one example we conducted is on the glass datasets by the TIA Center. So For a bounding box model, you need to provide many bounding boxes. And for the text prompting, you just need to provide a text prompt. And if you compare like the number of operations, so goals here is like a number of operations by the human. If you increase this like SAM and SM, the dice score, the segmentation performance will increase as you. So this is, let's say you try to have a bounding box for all the structures and hope it segment out. It will be very not very accurate, but you need to precise increase your human operations and increase the segmentation performance. And for the BiomedParse, we just need to provide like one manual input for the whole data sets. And this is also, excuse me. It's also a one model for all, so you don't need to train specialist models. And you can also, by iterative prompting, like multiple things in one image and resolving the conflict in the overlapping regions, you are able to conduct like multiple object type segmentation. So some of the applications for this, including, let's say, size measurement. So let's say after you segment out let's say the nodule, you can measure directly. So with what's the size of the nodule and like the size of the tumor and what's the ratio of like the enhancing part of the tumor. And you can also do like locating and counting. For example, you can find here are the like two nodules. There's the one nodule. With some like post-processing, you can split the segmented like a nucleus into like a different instances and then count how many of them are there. And we conducted like a so-called like a zero-shot adaptation. This is like the out-of-domain image provided by the Providence Hospital. They provided us like a few like pathology slides and have the model just conducted the segmentation. So in our training data, our definition is like neoplastic cells, inflammatory cells, and connective tissue cells. So on the pathology side, so initially they just conducted like a tissue level segmentation. It's very rough. They circle out when they gave us the image, they circle out, hey, here's the tumor region, here's like the lymphocyte and stroma, and without any like fine-grained cell segmentation. And then we further ask, can you provide like more detail? And then they kind of like circled out like these cells. And but with our model, we just need to provide kind of like link the tumor region to the neoplastic cells, lymphocyte to the inflammatory cells, and then have the model output similar like segmentation performance. There are a lot of areas where it's actually making a mistake. For example, let's say some of the cell types can look very similar to others when it's like going through like different cell in the different part of the cell cycle. But for the most case, it's matching the pathologist annotation. So because we train model on our collection of data, it can do things well within this region. However, we prompted with something that it never seen before. For example, like a different cell types or like a different type of lesions. It won't do it because it simply never learned it. So in that case, the model is able to fine tune to new tasks. So we conducted some like experiments with the so-called like a 10 shot fine tuning. So just to provide like 10 examples or 10 scans for the model, and then for the model to continue learn on it. And we found like the model is able to quickly pick up the performance comparing to like with a precise bounding box method. And so here is like a brief recap, but I'll have like some more technical details in the following. So BiomedParse is a text-to segmentation model for biomedical image with solid performance and able to do detection by identifying the prompts. in the image, and it can recognize like things in the image, and it's useful for with minimal user input and able to handle like multiple modalities. And however, there are some challenges remains. So I listed out a few challenges. The first one is highlighted because I'm going to address how we went further improved upon that. First one is locating the object purely by text prompt is not easy, especially when the object is small. Second is our model is a 2D model. So for most cases like a pathology slides and X-ray is okay. However, the 3D modalities such as CT and MRI, so naturally they have the 3D context, and our model is not able to utilize those contexts. It can perform well with a 2D slice of image, but still it's like underperforming when comparing to like the pure 3D context model. And it requires extra steps to go from semantic to instance segmentation. It cannot output, let's say, this is cell 1, 2, 3, 4, 5. It can output like a segmentation mask for all the cells of the same type, but you need to further do some post-processing to go to instance segmentation. And also same to the previous question, it cannot handle the whole slide image. You need to cut into very small crops. That's because the image is just so large. So I saw there is a question or not.
I think we have a few questions in the chat, Theodore, but I was thinking that maybe we'd save them for the end now.
Okay.
That's okay with you.
Yeah, that's okay. Okay, so for the last part, I will just briefly cover how we try to tackle the small object enhancement. So this is like after the BiomedParse initial version, we studied how we can further improve the model architecture. So let's take like one example, like what is the small object look like? So on the right side, this is a CT scan of the chest. And the target is try to find where is the nodule or if there is any nodule. So it can be very small, but the red region, the red circle is the ground truth nodule. It's very, very tiny. And this is actually a error case by BiomedParse, which outputs the green dot, which is another very small dot in the CT scan that looks very similar to a nodule, but it's actually not. So in some sense, it's just a missed the true nodule. So we call it like the kind of like the needle in a haystack problem. The object of interest can be smaller than maybe 0.1% of the image, and sometimes in this case it is even smaller. So if you look at how usually in transformer the tension mechanism is working, you calculate the tension score, let's say from any query to all those like patches, you will find out the tension score is divided by the summation of all these other features. So if the only relevant features is a very tiny portion, so it's very common that you will have a much lower like a tension score on the true relevant region if the portion is just so small. And this can be very hard for the model to focus on. So we devised a new attention mechanism called like the Boltzmann attention sampling. So instead of like having the model attending to all the image features, we do a random sampling first and then attend to the sampled patches. So let's say on this like 2D image, we first estimate a probability distribution of like where is the most likely region for me to focus on. And then we first subsample, let's say less than 10% of these patches and have the model only attend to these patches. So this way we can avoid this big number on the denominator because we don't have, we restrict our attention on a much smaller region. But what if like this smaller region doesn't contain any of the relevant features? So that's fine because we have multiple layers of the model, and we can refine the guess throughout the layers. So as long as like your this sampling hits one of the feature that's like more relevant, you can gradually refine this like a probability distribution such that you can sample the right features that you want. And in order to do that, we encourage the model to do more exploration in the earlier layers and exploitation in the later layers. So here's an example of the sampling. So in layer 2, the model is like a basically random sampling across like on this like a CT scan. So this red region is the ground truth nodule. So it has samples there, but also exploring other regions. So when the model goes to like a middle layer, like layer 5, you can see it's like more concentrated in this like target region, but still exploring other parts. And in the later layer, layer 8, is mostly concentrated on this region. So throughout the layers, this gas is getting improved by the feedbacks throughout the layers. And another example is trying to find the pancreas in the abdomen MRI. So in the earlier layer 2, none of the features were sampled from the pancreas. So in some sense, if it's not sampled, the model never have to see it. So in layer 2, the model doesn't even know how this pancreas looks like. However, in the middle layer, layer 5, some of the samples like locate on this correct region. And because of like the signals from here is enough for the model to pick up, hey, these are more relevant. So in the later layer, the model is able to concentrate this probability distribution and mostly like exclusively sampling from this target region. So by putting this Boltzmann attention sampling into the whole model architecture, we build like this like a Boltzformer architecture. So this is also to the question of the model architecture in more details. So this graph shows like the more detailed model architecture of the original BiomedParse architecture and the new implementation. So the only difference is like we replace the regular like cross attention mechanism with like this Boltzmann attention sampling mechanism, and everything else stay the same. And by doing so, we observed that this like Boltzmann sampling methods actually outperform the full attention mechanism, where you just have the regular attention on the full image, and that way it's very hard for the model to pick up those like small objects. And with this like dynamical sampling, it's also outperforming some like a fixed threshold method. And we compared the performance advantage on the small objects, which means the object size is less than 1% of the image size, and also on the large objects. And we observed that the advantage on the small object is much larger than the advantage on the larger objects. So on the larger objects, the Boltzformer architecture versus the SEAM architecture is almost the same. There are no like a statistically significant advantage. But on the small object, the advantage is very clear. So here is like the small object example. So the original architecture, the nodule is missed, but with the improved architecture, the model is able to precisely locate like this very tiny nodule. So I guess yeah, that's all for the presentation. I think also we are good on time, and thank you so much for the attention, and I'm happy to take the questions here.
Well, thank you so much, Theodore, for a really fantastic talk. And as I'm sure you've noticed, there's lots of questions already in the chat. So yeah. I will start to ask some of these questions. So Kezi from the TIA Centre has asked, how robust are the segmentation results when faced with different descriptions of the desired mask? Is there any ambiguity in language prompts used to describe the desired segmentation region?
Yeah. So we actually conducted the ablation study. So for every segmentation task, the LLM output like a variety of different text prompts. So we conducted like two evaluation mode. One is like randomly sample a prompt from there. The other is like purely describing in the standard format. Let's say the object type, basically, let's say. The other like the standard description, what's the object type in what the site and modality in the standard manner. So the two evaluation, the performance is not it's almost the same. It's like there's no statistical difference between like a prompting the standard manner or prompting slightly variation. But it can be a bit more variation. So let's say if you prompt describe things in like different the object type have a different terminology. Or for example, let's say the model learns mostly the neoplastic cells, but if you ask for tumor cells, it can kind of understand it, but sometimes there are like even more if you ask for like a lesion. So sometimes the concepts they are like subsets and or parent concepts. The model can be confused. So if you describe in the exact very precise manner, it's able to locate it. But if you prompt in like more like vague manner, sometimes it can not understanding very nicely. So that's why in the original architecture we also have like a GPT-4 LM try to help the model to navigate like those like more like out of the distribution description. But no, most of the cases it's okay to handle different type of prompting.
Sure. Thank you, Theodore. And I hope that's answered your question, Kesley. Next, we've got a question from David Epstein in our group. He's asked, does the LLM know before you start that different text descriptions describe the same medical concept? Or do you have to train the LLM to make these identifications?
We didn't train the LM, so we used the GPT-4 at the time just out of the box, and we were not able to further train it. But it's a very nice thing just doing the variation because all the concepts actually are presented here. So you just need to piece those like text prompts in different ways. And we did a manual check of the text prompt to validate like if it's like actually hallucinating like different if non-existing like object types or modalities and filter out all those like prompts. But yeah, we didn't train the model.
Thank you. And we've still got a couple more questions in the chat. So Craig Miles has asked, said the unified approach is really nice. Does the inclusion of multiple imaging modalities lead to superior model performance compared to models trained exclusively on a single modality, such as pathology?
Yeah, I think it's a very good question. So so for example, let's say in CT and the MRI, sometimes they can look very similar because they are all doing the scan of the, let's say, the same body part. Let me see if I can find an example. So yeah, so this is CT and this is MRI, although like this is in the chest region, this is in the abdomen. But if they are both conducted in the abdomen, they can look very similar. So and all those organs, they share the similar shape. So I believe the model is able to leverage like more the information across multiple data sets to try to learn better. So yeah, I think that's true. And some modalities, they can look very drastically different. Let's say in ultrasound, the liver looks very different from ultrasound in the MRI. So that way, like my personal intuition is like maybe the model is not able to leverage the similarity there, but for let's say CT and MRI, I believe the model is able to try to learn better by those information across different modalities.
Oh, thank you. I think what Craig was also asking his question was, have you actually compared? So, for instance, if we take pathology, and you looked at, obviously you mentioned PANU. Did you manage to compare, say, the detection results on, sorry, the segmentation results for different nuclei compared to, say, state-of-the-art methods such as HoverNet or StarDist or something like that?
Yeah, we didn't do the detection because one of the limitations of the challenges is actually the model is doing semantic segmentation rather than instance segmentation. So you need to further go through like doing instance segmentation using another post-processing. For example, like using HoverNet can help kind of like make the semantic going into instance segmentation. But because not the core model capability, so we didn't do a by instance detection comparison.
Yeah, I suppose what I meant by that was if you took, for instance, the ground truth, like the centroid, and used that as a prompt, or well, not necessarily that as a prompt, but because obviously it's text based, but yeah, okay, thank you. And we've got a final question online, and then before I ask everyone people in the room, so Pingjun Chen has also asked, do you consider generating pathology slide patches to have different magnifications? So how do you consider different resolutions of radiological images also? So a question about the resolution.
Yeah. It's a very, very good question. So actually we just the data set we got is like a fixed in this size. So we didn't further enlarge it, and we could not like make it like or zoom out. So we just used a fixed resolution for this specific one. So for example, when we conducted the segmentation on this like a bigger slide, is still not whole slide image, but it's much bigger. We manually cropped it into many crops and have the model scan through those crops and perform segmentation and glue them together. So if you look closer, you can actually see there are some like a disconnection on the border sometimes, but it's like it's very small. So I think like a different magnification is a very important it's a very important capability, because we try to handle many different modalities at the same time. It doesn't make too much sense to say if you zoom into a X-ray or zoom into a CT, that make the image a lot different. But yeah, I agree for the like these like pathology slides, it makes total sense to have different magnifications. But when we train the model using the dataset, we didn't do that step.
Thanks, Theodore. I know we started this seminar 5 or 6 minutes late, so do we have any final questions from anyone online or in the room that would like to finish the seminar with?
Just a quick one.
Yeah, of course. So we've got a question from us in the room.
No one has. Actually, I have two. So first of all, thank you very much, Theodore, for the fantastic talk. I really enjoyed your work. So my question is, can you remind me which data sets did you use from pathology in your work? That was Panu, was GLASS?
Yeah. So for pathology, we just used like these two data sets to handle this. Yeah.
Yeah. I mean, I understand your work is limited to semantic segmentation, which in my opinion, like in terms of nuclear detection, I think right now the challenge is actually in the semantic part, meaning that state-of-the-art models, they do not do a good job in classifying cells, right? They do a good job in segmenting them. So I was wondering if you could have or had already investigated that with your model. Say, have you tested how would you semantically segment a certain class in different data sets? So there are several data sets available for cell segmentation. And just wonder, how does your model, perform when there is a shift in the domain, like there is a domain shift, and how does it generalize well or not?
Yeah. It's a very good question. So we didn't do like a quantitative comparison when the domain shift, but we did do, let's say, so these are like slides from the hospital. So they are not in the same distribution as PNUC, but the model is able to do a okay job on that. But we didn't further do, because the pathologists, they didn't have like a very fine-grained annotation for us to do a quantitative comparison. So we didn't have the quantitative results. So we just manually have the pathologist check the results. Yeah, but I agree. There are a lot of other like data sets that's for you to compare across different like the domain shifts. But yeah, for this work we didn't cover that.
Yeah, I understand, because this is a huge work. You couldn't do everything, of course, in just one work. But like if you're thinking to further, expand this and investigate your new idea, it would be really nice to also see that in our field, just as a suggestion.
Yes.
Just one other quick question.
One short quick question.
Very short question. So I know you said that one of your major challenges was the dataset curation, which is completely right. So what was your next hard challenge to overcome? What did you find really hard in your project? What would you do differently if you go back?
I think like so that's was one part. Also like a training the model making it able to conduct that segmentation is not that straightforward. So you need to first like make sure your text prompt is good in providing the different information for the model to conduct different tasks. So there are a lot of like analysis of the results and also modalities because the image modalities is changing and also the text prompting is changing. So yeah, maybe one example is MRI. So MRI have many different sequences. They all look very, very different. Some of the object types, let's say some parts of the tumor is not even visible or not, I cannot say visible, it's not visually distinguishable in some of the modalities. So you need to go to other modalities for model, even for human to recognize that. So that poses a very significant challenge for the model because however we train model is just cannot conduct certain tasks in some certain modalities. So we did some like a manual analysis and clean the data sets such that all those like object types, they are visually distinguishable in some certain modalities. And then this is just one example that this like modality shift actually is a bit challenging when you hope one model to solve everything.
Makes sense, yeah. So data set and compute, basically. Well,
Yeah,
thank you very much again.
Yeah.
Brilliant. Well, thanks, Jasper.
Thank you.
And thank you, Theodore, for again a really fantastic tour. So thank you for sparing the time. And yeah, good luck with your future work. And before we finish the seminar, just a quick reminder to everyone else, our next seminar is in 2 weeks' time on Monday, the 12th of May. It will be at the usual time of 2 till 3 p.m. We'll be joined online by Professor Jakob Kaffer from TU Dresden to discuss their lab's work on agental AI in pathology. So I hope we see you all there. And also on the same day, the 12th of May, is the abstract deadline for our special session in Frontiers in Contextual Pathology at MIUA. So 2 weeks until the deadline and 2 weeks until Professor Kaffer's talk. Brilliant. Well, thanks again, Theodore, and thank you everyone for joining us online and in person.
Yeah, thank you so much. Thank you
Thanks,
for hosting.
Theodore.
Thank you.
Bye.
Yeah, bye.